Welcome everyone to our fifth event in Contact's Produce Safety webinar series. This series tackles a different fresh produce safety topic every month through a dynamic webinar series that brings together industry, academic, and regulatory minds to solve some of the produce industry's toughest and most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, where scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by a specialty crops research initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. This webinar series, coupled with other research and extension efforts, is part of a collaborative effort across 10 different institutions to enhance produce safety knowledge and compliance across the industry. We are so grateful to our collaborators for their efforts and support in and of this webinar series. You can see their logos and as well as our website on the slide. This webinar series will continue next month with a talk about building and implementing effective environmental monitoring programs on Thursday, April 14th, 2022. I am very excited to see this webinar as the team will present a very neat and relevant and timely case study at the end about sanitation research that was driven directly by industry need. This multi-year webinar series continues through May, and we will wrap up our first season with supply chain integrity. We will break for summer and return in the fall with even more industry proposed ideas. If there are topics that you want to hear more about, let us know in the comments, webinar surveys, or on our social media pages. Again, this is truly driven by the audience and by all of you. About today's webinar or other produce safety news, research, and education on our Twitter and Instagram pages. Please follow us on these platforms at FSCI for Produce Safety Science. So let's welcome our speakers today. We have Billy Mitchell. Billy Mitchell is the Food Safety Modernization Act, or FSMA, training coordinator at the National Farmers Union and works closely with the local Food Safety Collaborative, an FDA-funded initiative to provide training, education, and technical assistance to local food producers. A former farmer, cookie baker, chef, West Indian drum and dance therapy assistant, composer, and bus driver drummer for a band that involved a lot of face paint. I feel there's a story there. He has now the opportunity to work alongside producers and other educators all over the country to identify creative and cost-effective solutions that make food safety practical for small and medium-sized operations. It's an amazing job, and he's thankful to be surrounded and supported by so many wonderful people in the food safety and farming community. Joining him will also be Dr. Dave Ingram. He is a consumer safety officer with the produce safety staff in the Office of Food Safety. He is interested in the mitigation of foodborne pathogen prevalence and persistence through irrigation water management and disinfection practices and the application of various practices for the treatment and handling of soil amendments. Dr. Ingram's research has been instrumental in the development of the United States Composting Council, USCC, testing standards for human pathogens in finished compost, and his work on the fate of human microbial pathogens during the production of compost and compost teas was used to develop compost tea regulations as stipulated by the National Organic Standard Board. He received his BS degree in biology from Dickerson College and both MS and PhD degrees from the University of Maryland at College Park. The last of the speakers will be Donna Clements. Donna has worked with the Produce Safety Alliance since 2015, first as the Southwest Regional Extension Associate, and now as the PSA Coordinator. Donna's role with PSA is to offer training courses on the FSMA Produce Safety Rule, provide technical assistance to growers and produce safety educators, and develop educational food safety resources. In addition to outreach on the FSMA Produce Safety Rule, Donna's specialty within the PSA has been developing resources on biological soil amendments of animal origin and sanitizer use. Donna's involvement in the fresh produce industry began at a young age while working on her family's fruit and vegetable farm. She has experience in production, harvesting, post-harvest handling, and sales of perishable products. Donna received both her Bachelor of Science and Master of Plant Science from the University of Maryland. Welcome all. The floor is yours to start.
Oh my gosh. Thank you, Laura, for those introductions. Thank you, Donna, for sharing screens. And yeah, I think we can go to the next slide and kick it off. So this is just one of my favorite photos. This is an action shot of me and a colleague going to grab another wheelbarrow of compost as we filled what felt like hundreds of raised beds in an urban farm in downtown Atlanta. And having Laura list off some of my non-food safety bona fides, some of my past experiences, was in no way an attempt for me to overcompensate compared to Donna and Dave's bios or try and prove that I don't have imposter syndrome. But even if it was also a way to show that we just have very different backgrounds and we get to work with farms of very different backgrounds. Farms used to working with the government, farms that have never worked with the government, farms that do a ton of work with extension, farms that don't. And it's been amazing to share our experiences, our different backgrounds in research, education, being on farms, and what we've been seeing and what we've been hearing on farms. So how did we get to this webinar? A few months back, I had a chance to be on just this beautiful small scale and covered, as in covered by the Produce Safety Rule farm. And they asked me about spraying an agricultural tea made with compost that they'd made on their farm using a scientifically validated process for a foliar application. So they wanted to spray it on their crops, and they wanted to mix in some extra goodies, some additives like kelp and molasses. And they really wanted to know, okay or not, with the produce safety rule. And I told them not with all those additives, the kelp and molasses, spraying it all over the crops, but maybe as long as the compost had been made using a scientifically validated process and it was just compost and it was the water, I thought probably okay to spray and apply as a foliar application, but I like to live a low risk life. And so I said, let me ask some people that are much smarter than me. And so I emailed Dave and Donna about it and just opened up a whole can of worms. And so we've had a ton of emails, great conversations, this generous offer from the contact team to share with y'all what we've been discussing, what we've been learning, along with a lot of other great produce safety friends and pals. And we've all ended up with y'all here today on this webinar. And I certainly have a much better understanding of the risks and the rewards that come with agricultural teas because of all the work we've been able to do the past few months. And I hope that y'all leave today with a better understanding as well. So today we've got some science, we've got some interpretations, and we've got just like a real desire for y'all to share as many questions as you can. And we need to understand what they're doing and how they're using them so that we can offer the best guidance and do the best research possible. So that's why we're here today. We're going to offer up some definitions. We're going to talk about why and how growers use agricultural teas. We're going to talk about the risks involved. We're going to touch very briefly on the NOP, on organic farming. So with that, Donna, let's go to our first definition slide. So we've got the three Bs, three big B definitions that we really should know. Biological soil amendments, biological soil amendments of animal origin. Thank you, Laura, earlier. It is pronounced BSAAO, and then biosolids. Biological soil amendments are any soil amendment containing biological materials. So this is compost, manure, blood meal, feather meal, peat moss, pre-consumer vegetative waste, your big bag of leaves, yard trimmings. That's a biological soil amendment. Biological soil amendments of animal origin, BSAAOs, are biological soil amendments which consist in whole or in part of materials of animal origin. So we're thinking like manure, blood meal, feather meal, mortality composting, and also includes table waste. What it does not include is any form of human waste. And that kind of gets us to biosolids. Biosolids are a product of the wastewater treatment process. Oftentimes biosolids and sewer sludge are used interchangeably. And we should just be super clear. You may not use untreated human waste. You can use biosolids. The application is limited to using Class A biosolids. I will say all these definitions, we're going to go through real quick. So y'all, if you haven't taken the Produce Safety Alliance grower training yet, we'd really encourage you to do that. Next slide, please. With agricultural teas and soil amendments, there's a couple of commonly used and confused terms. Those terms are leachate, aqueous extract, agricultural tea, and agricultural tea additive. Leachate is this liquid that passes through organic material, and it can contain pathogens, phytotoxins, anaerobic microorganisms. This is a beautiful compost pile. And probably leaking out the bottom of it is this smelly liquid. It used to be my job every morning to take a push broom and push leachate off a compost pad or off a concrete pad where we're making compost, because it just smells. That's a liquid that you do not want to be spraying on your crops. I actually found this great NC State Master Gardener publication that says leachate can be poured down a toilet. So that was their recommendation. Aqueous extract is a water extract of biological soil amendments that's been held for less than an hour before application. An agricultural tea is a water extract of biological soil amendments that's been held longer for 1 hour before application. And agricultural teas are a soil amendment. Agricultural tea additives are something that gets added in. That could be like molasses, a yeast extract, humic acid, kelp, and they're really added to agricultural teas to increase microbial biomass. Next slide, please, Donna. So that aqueous extract, the big thing is less than 1 hour. If you've mixed it up and it's been less than 1 hour, it's an aqueous extract. Our big takeaway here is timing is everything. Once it's been brewing for more than an hour, it's reclassified as an agricultural tea. And that's really because we've given it some time for things to happen. Next slide, please, Donna. And so y'all, this is a brief and super high level overview of a scientifically validated process. And we're pulling things from the FSMA, Food Safety Modernization Act, Produce Safety Rule. The first one we're going to touch on is 112.54 nuclear bomb proof. Okay, so that quotation mark thing, that is not in the Produce Safety Rule, but that is something I've heard Dave Ingram say a lot. And so he might say it today. So we thought we would just introduce it right now. And if it's followed that process, you can apply it in any manner. And it's really like Listeria monocytogenes, not detected. Salmonella species, not detected. E. coli 0157H7, not detected. What we really see on farms, and that farm that I got to work with and talk about compost tea a few months ago, is 112.54B1 and 2. One is static composting. That's a minimum of 131 degrees Fahrenheit for 3 consecutive days plus adequate curing. The other one is turned composting. That's a minimum of 131 degrees Fahrenheit for 15 days with at least 5 turnings and adequate curing. This is an aerobic process. You might be wondering what's adequate curing, and that's just like a whole nother webinar. And the rule for that is you need to minimize the potential for contact with covered produce. Donna, if you'll go to the next slide, please. So why are people using agricultural teas? So if you ask my really good friend Max here, who's showing off his compost tea vortex brewer, he'll tell you that they were doing it because they wanted to improve soil health. They wanted to improve plant health, and it was just like more bang for their buck. You have a certain amount of compost, you can spread it over a limited area, or you can mix it into this water, you can aerate it, you can bubble it, and you can spray it all over the place. And Max and a lot of growers believed that it was going to improve soil health, improve plant health. And we've found that like maybe the jury is out. We need more research. We need more evidence. And so I will say not everyone agrees with my friend Max. And there's a couple websites, and we will share more links after this webinar. But Dave showed us this one website, The Myth of Compost Tea. So if you look this up, The Myth of Compost Tea, there's some just pretty interesting articles. And there's even this one from University of Illinois Extension, that I think has snake oil in the title. And so you can get some different perspectives, because there is a ton of popular press around compost tea. And that's where a lot of growers are getting this information from. But we really could use more research around it as well. So We know why they're doing it. They're trying to improve their farms. They're trying to improve their soil health. How are they doing it? So you can do it through drip irrigation or injection. So putting it through your irrigation, mixing it with your water. You can do a soil drench in a bunch of different ways. That top picture is a small scale farmer. You can see them, they're drenching their soil with an agricultural tea. A lot of growers will put it into backpack sprayers and spray it around, or you can put it into a sprayer truck so you can apply it to really large areas. And something that a lot of growers are doing is foliar application. So they're taking the agricultural tea and they're spraying it onto the plants themselves, the stems, the leaves, the flowers, the harvestable portion of the plant. And there is, I would say, a pretty commonly held belief that if you do this a couple of weeks before harvest, that it's going to bring about a ton of benefits. And there's not often a discussion about whether or not there are those rewards. What are the potential risks that can come with that foliar application? Next slide, please.
Okay, so I believe this is where I'm going to pick this up. Thank you, Billy, for that introduction. So I do want to take a second, and now that Billy talked a little bit about why compost tea is being used, or agricultural tea, I wanted to talk a little bit about the how is this being brewed and what are some of the key factors to consider when we talk about risk. So looking here, there are a few primary ways that we see agricultural tea being brewed on the farm. Starting with, I'm going to say the most simple method is what is usually called the bucket method. So this corresponds with the top photo on the right. And you can essentially see this is pretty much a tea bag made out of burlap. There is treated compost in that tea bag. And essentially it's made by immersing that sack or that burlap sack of compost in water. And usually this method is done over a series of days to weeks. It's a passive method, but very low input, very simple. The second brewing process is what we call the bucket bubbler method. And this is where we start to take that method. We have compost immersed in water. And what we're adding here is essentially aeration. So we're aerating it. And by aerating the tea, we're going to talk a little bit more about this later, but by aerating the tea, you're really trying to brew those beneficial microorganisms, and adding that aeration shortens the amount of time that we're seeing the agricultural tea being brewed. So the intent here is it's aerated. You can see in this photo, it's adding in that oxygen, and then the grower here would use this immediately. They would take it out immediately and use it on their operation. Then the third method that we often see, this is more of a commercial method, so this is going to be larger farms, is called the trough method. And instead of being done in a bucket, we see recirculated, the compost tea or the agricultural tea is recirculated. So looking at this photo here on the right, we see these little lines. They have water, and it's essentially, there's compost. Sometimes we see the compost in a trough, like a PVC pipe. But the idea here is that the water is recirculated. Again, this is usually over a series of a couple of days. And by recirculating, it's just going through that tea and continually leaching out those supposed benefits. So I wanted to, because we're going to turn this conversation to risk, I wanted to note a couple of primary brew factors, is what I called them. The first one is the feedstock. And I have a question here is, what is the feedstock made out of? Generally, because this is compost, has the biological soil amendment of animal origin gone through a validated treatment process? And Billy outlined what the two standards are in the FSMA Produce Safety Rule a little bit earlier. So that's the first question. Has it gone through a validated process? And what level is it treated to? Has it gone through a windrow composting or an aerated static composting? Or has it gone through, I think what Billy called nuclear bomb proof? So has it gone through that other method where it's been like a heat treated poultry pellet or something like that? That's been gone through a process. The second factor I want to introduce is aeration, because we see the introduction of oxygen having an impact on pathogen. And then the last factor is, what about additives? And this is something, these factors Dave is really going to highlight when he presentation. So looking at aeration, the main reason we're talking here is that aeration is encouraging the growth of aerobic microorganisms. If you think of turning a compost pile, so if you step away from agricultural tea for a moment, think about taking that shovel out, turning the compost pile. What we're seeing here is a flush of oxygen aeration, and it's really encouraging growth of those aerobic microorganisms, which are generally seen to be biological or the beneficial microorganisms is what we're trying to really brew in these agricultural teas. Aeration shortens the brewing time of the tea. So instead of seeing tea brewing for days to 2 weeks to 3 weeks is usually what we see, it's generally down to 18 to 36 hours. And again, Dave is going to talk about a little bit of the research that we see about aerated agricultural tea versus non-aerated agricultural tea, which is that passive process. I also wanted to introduce this discussion of additives because coming into this conversation that we've been having with Billy and Dave, I would always hear this question of additives, and I would hear people say, we're adding this additive to the agricultural tea. It's really all about the entire process. It's not just adding compost and water or adding compost, water, and aeration. It's this whole package process of additives are really part of this process. And what we're intending to do is brew up beneficial microorganisms is the intent that we see here on farms. So additives are sold as a microbial starter. Oftentimes we hear about molasses being used, and that's intended to be a quick, easy nutrient source for these microorganisms. It's intended to encourage a rapid increase in concentration, metabolic activities. And I loved on the right-hand side, you can see there's an additive here called microbe multiplier, which I thought was a great name. And I wanted to point out this ingredient list because it's not only molasses that we're seeing. There is research out there. Dave's going to talk a little bit about this, but there is some research back from the early 2000s that shows, look, molasses, when applied to agricultural tea and brewed in that manner, it increased the regrowth of specifically E. coli O157H7 and salmonella. They also found that the natural microbial community present in the agricultural tea did not prevent pathogen regrowth. So I know when we do grower training courses, we often get the question of molasses. So molasses is, I think, a pretty easy example to say, not a good idea. Don't add molasses to your agricultural tea when it's being brewed. But what about some of these other ingredients that we see? So on the right-hand side here, this product has alfalfa meal, azomite, frass. Frass is insect poop for those who are not familiar with that term. But I wanted to point out humic acid, kelp meal, because those are two of the examples that I often hear about. And I just want to highlight, Dave's going to go more into detail here, but I want to highlight that there are studies looking at pathogen regrowth in additives that are not molasses, that are not that really carbon-rich source, and they still stimulated pathogen regrowth, even though it was humic acid, kelp meal. And those products are really intended to amend the physical characteristics of the compost tea, not really serve as a starter. But I wanted to highlight that what we see in a lot of the published research is that pathogen regrowth is not readily observed in teas made without additives. Okay, so as I prepare to turn this over to Dave, I just want to highlight what we know so far is where do the risks lie? So a quick reminder about risk. The definition of a risk is the probability of an adverse health effect and the severity of that effect. So we know that taking compost, brewing agricultural tea, it could be hazardous. So there is a potential hazard there. But what we're trying to do with this talk is highlight what do we know? What are some of the factors to consider when brewing agricultural tea, when we're applying it? So is that agricultural tea contacting the harvestable portion of the crop? We know that when there's direct contact, we talk a lot about this in agricultural water. When there's direct contact, there is a potential for the direct transfer of pathogens. And we're going to talk a little bit more about biofilms later, but biofilms can protect the pathogen on the plant surface. So that's something else to consider. And I want to highlight quickly some previous USDA guidance that is published. This is from 2006. And this is the National Organic Standards recommendation for guidance, use of compost, vermicompost, processed mort, and compost tea. And what this older guidance document highlights is these following recommendations. So when we're talking about compost tea, which we're calling agricultural tea, it must be made with potable water, sanitize equipment before use. So we're getting to see some basic good agricultural practices here. And then I want to highlight these other two bullets. Compost tea made without compost tea additives can be applied without restriction. And compost tea made with additives, like the ones I highlighted earlier, can be applied if it's tested for indicator bacteria, the recreational water standards, or if restricted to a 90-120 day interval. But I'm going to pass this over to Dave to talk a little bit about how some of this guidance has evolved over time.
Thank you so much, Donna. This is excellent. You guys covered so much information, and I'm so happy to be here. This is very much in line with our Educate Before We Regulate mission. So we've been walking around the country doing a lot of these soil amendment workshops, and invariably agricultural tea comes up, and there's just so little time in those workshops to give this marvelous agricultural input justice that this is just a wonderful way for us to expand on some of the concepts. Education and before we regulate is our motto, and this is why we're doing it. But really not just doing these presentations for you guys, this is just as much for FDA. So at any time, and I see a lot of you in the audience I don't recognize, I'll give a little background of who we are and what we do. But at any time you see something that you don't like, or you see a process or something I say is untenable in your growing operation, I really would appreciate you reaching out directly to my office. My email is on the bottom of my picture, and let's have a discussion about it, because what I'm going to talk about today, a lot about subpart F, is still under the pen. We're not finished with that regulation yet. We still have a few years to get our regulations correct and make sure that feasibility is also in line with the safety that FDA expects to come out of these regulations. So without further ado, I want to give a shout out to our whole institution here. This building behind you see is our home office. We've all been hovering around it for the last 2 years, but this is our home base. We really refer to this as the F in FDA. We are the food safety people, but we also do a lot of other things besides foods. We regulate cosmetic products. We conduct a lot of good science. And we do honest labeling programs. We do so many things to make sure that the food you eat every day are safe. And this really predates a lot of the federal regulations that you've seen lately. But the Food, Drug, and Cosmetic Act has been around for a long time. So that is our grandfather clause that we always refer to when we see something that doesn't fit nicely and neatly into our new regulations. We always fall back to the Food, Drug, and Cosmetic Act. So bottom line, we want to make sure the food that is grown on the farms, that is shipped, processed, packaged, as well as imports and domestically produced, are safe for you to consume. So I'm a FSMA hire. I came to FDA in 2013, right when the Produce Safety Rule was drafted, came out. I came over from the USDA. As Laura mentioned in my bio, I spent many years, probably 15 years in USDA, working on agricultural issues on the farm. So when FDA came and decided to regulate, it was important, Congress decided it was important to set FSMA up. And the Produce Safety Rule is one of the major pillars of FSMA. We started to regulate how growers started to do certain things that they've been doing for a long time. I was very fortunate to be one of the hires to help navigate the science to make sure that there is good science behind the Produce Safety Rule. And the Produce Safety Rule is divided up into chapters. Those chapters are subparts, and each subpart is dedicated to one of these areas that we all recognize as probably could be potentially hazardous to the production of safe food. And soil amendments is where agricultural tea fits. Why? Because agricultural tea used to be called compost tea, and compost is a soil amendment. So it naturally fit nicely and neatly into subpart F, which I helped to morph into what it is today. But as I mentioned, subpart F is not finished. And that is all about how we regulate and utilize, how we decide to help growers utilize soil amendments safely. So the Produce Safety Rule obviously is designed to act as a firewall to help prevent contamination through all these routes. And one of the questions I always get is, well, okay, boy, that's a lot of stuff FDA is regulating. What doesn't FDA regulate? What's not in the Produce Safety Rule? There's a lot that's not in the Produce Safety Rule. For example, if you're using a soil amendment that's not of animal origin, it's not beholden to subpart F. And it's difficult for me, especially coming from an agricultural compost tea background and a compost production environment, I know that all compost has the potential to contain foodborne pathogens. So when people ask me what's not regulated under Produce Safety Rule, oh, I'm making compost with strictly vegetative material that's not regulated by the Produce Safety Rule, so I don't need to look at your regulations. I talk about the science, and I talk about our rational thinking behind setting up Subpart F and why all compost should be included, although we're not able to write up violations that are associated with soil amendments not of animal origin. It doesn't mean growers don't have a responsibility to look at the rule, understand the rule, and that's why we're out here to help people recognize the potential hazards with certain practices. So this is a personal story for me, and it's personal because I come from a medical microbiology background. My first degree was in medical microbiology, and I work with some of the very big names in the industry, one of which he was a captain in the Navy and worked with all kinds of enteric diseases around the world and has done so much good to prevent enteric diseases. I fell in love with understanding how these foodborne pathogens could just be destructive to the human biology and how they interact. And I was fascinated with recovering and isolating and understanding these pathogens. And my master's degree involved developing a rapid diagnostic test to look for 015787 in ground beef, because back in those days, in the 90s, ground beef was the hazardous source of 015787. So those of you remember back in those days, we didn't have fancy things like whole genome sequencing. We didn't have metagenomic research to help us understand how to grow these organisms better or understand the microbiological communities. We had agar. We had most probable number techniques. We had what you all do in the science probably regard as the dinosaur. I'm probably a dinosaur in the parlance of your times, but I look back in those days and it gave me a great perspective. I was taught the many ways human pathogens are able to adapt to the human biology to get the nutrients that they need. And I understand that when I recognize a human pathogen that I'm having trouble isolating and recovering and growing in the lab, the way we grow these organisms is throw more nutrients at them. We add more nutrients. We have wonderful agar plate medium that have a lot of nutrients. And if the organism is not growing well, we add more nutrients. A great example is blood agar. Yes, sheep's blood. We add those to organisms that have hemolysins that break down those blood cells to get the nutrients for the organisms to grow. And if that's not enough, we cook that blood agar, turn it into what's called chocolate agar, which automatically releases all the nutrients in the agar. So we have more nutrients for the pathogens to grow. Fastidious organisms grow on these plates that don't have the capability to get nutrients other ways. And we even have what's called brain heart infusion agar. I'm telling you these examples because my mindset is when you can't grow a human pathogen in the sample, you put more nutrients and more nutrients. So that's my mindset. When I moved to the USDA to help run the compost research facility, I started to learn about soil health. And I worked in the soil microbial systems lab for a wonderful scientist, Dr. Patricia Milner, who taught me everything I know about composting. And I recognized that there was a wonderful lab in that building that designed a nutrient agar medium to grow rhizosphere. So when I came to that shop, I was just using the same media that I used to grow human pathogens because I was interested in the survival of human pathogens in soil and in composting process. And down the hall, the exact opposite was happening. This shop, under the guidance of Dr. Beyer, I think he's retired now, but he designed a medium that mimicked the nutrient profile of the rhizosphere. And believe me, there is no better steward of the soil health organisms than plants themselves. The plants' exudates that come out in the root zone of plants will grow every organism that they need to help extract nutrients from the soil. So the plants have a wonderful mixture of different and even hormones and other chemicals that shepherd the microbiological community that the plants need to grow very well. And so this lab designed an agar plate called rhizosphere isolation medium. You can look that one up, and it has a very complex nutrient structure, but very little nutrients. In fact, if you take a trypticase soil agar plate that many of you are familiar with, and you cut that into 1/10, that still has more nutrients than this isolation. And so when I started to use that agar plate to grow the soil microbial community, I would get 1,000 times more colonies on the plate. And so here's an example. It's just this eureka moment I had that I was perhaps looking at understanding the soil community a little differently. And this is a great example of when you recognize what you realize how little you knew before. So the more you know, the less you knew, if that makes any sense. But, and so here we go. Now my brain is starting to morph medical microbiology techniques with environmental technology isolation. And environmental microbiology and medical microbiology, they're two different worlds, and there's very little communication between the two. So I was out there fending for myself, and growing E. coli in these extreme environments really helped me to understand that relationship. Because in the middle, and I'm so happy every conference I go to now, the hot button item on everybody's lips, at least in the growing community, is soil health. How am I going to maintain my soil health? And you know what the best way to maintain soil health is, at least from my research perspective, is to maintain the microbiological community associated with soil. And there is no more complex way of adding the beneficial microbes to your soil than compost. Compost, and now FDA is regulating certain composts that are of animal origin. If you want to apply compost in certain ways, we're restricting you to making sure that you go through a PFRP process. So what is a PFRP process? PFRP is a process to further reduce pathogens. This is a time and temperature now that's codified by FDA Produce Safety Rule, by which if you go through that process, then you are able to use compost in a manner consistent with the Produce Safety Rule, which means you can apply that compost in a manner that minimizes contact with the produce. If there's produce in your field, let's say you have an established bed of leafy greens, you still can use compost to add a little nutrients to the leafy greens before harvest. But as long as you're not blasting this compost across the bed of leafy greens, you're not sprinkling this compost on the tomato crop, you are able to use compost to meet the nutrient demand. Now, granted, this is a little bit tricky to apply compost without blasting the compost itself, the produce itself, but you can use it because we know that compost has, if it has been made correctly, you have dramatically reduced the pathogen presence in that compost. And let's face it, every teaspoon of compost is going to have 10 to 100 billion organisms of beneficial organisms in there. And so the residual pathogens in that compost, after it's gone through its PFRP process, will not have a chance to survive for very long. Now, I will tell you that in our experience, compost is not pathogen-free, and it should not be considered pathogen-free. In fact, yes, we've done a lot of compost testing over the last 2 years, and we're very comfortable with the process of composting, even more so now because we haven't found any pathogens in those composts. But my personal graduate school experience, I looked at a lot of compost as well, and I would find residual pathogens in the compost. But the numbers of pathogens are so low, knowing that compost has gone through the proper process, and I would sample compost that have gone through a process, the proper composting process, they met the maturity index levels that we expect. Compost is stable, that it's not going to reheat in a Dewar heating flask. We walk through all the different parameters, and a great way to know that your compost is meeting these requirements is to get a compost that's certified by the Seal of Testing Assurance program within the United States Composting Council. It's a document disclosure program that every compost that meets those standards are beholden to provide you with a certification of all the tests that compost has gone through. And these are the composts that I sampled. And yes, I did find residual pathogens in the compost. We've done a lot of research since then with FDA to understand what the fate of those pathogens over time. And again, I'm not too excited about that because compost is the way to go to maintain soil health, plant vigor, suppress phytopathogen and human pathogens in that soil. I'm getting off topic here because I want to circle back to agricultural teas. Back in the day, when I was cutting my microbiological teeth, I learned about this practice of compost tea. And this is a very long long-standing process. This is a process that's been walked its way through Europe, probably even further back into Asia, but the European methods involve throwing compost in a bucket and stirring it up and let it sit for a week or two and applying it to your soil. Non-aerated compost tea. When I was looking at food safety issues for USDA back in 2000, even before in the mid 90s, probably American ingenuity came into play. Well, American growers, they don't want to wait 7 to 10 days. So what they do is they throw their compost, as you saw through Billy and Donna's presentation. We throw compost in a bucket and add oxygen. Oxygen is a great way to increase biomass very quickly. At least the aerobic organisms grow a lot faster than anaerobic organisms. And guess what? E. coli and Salmonella, they're facultatively anaerobic organisms, but that doesn't mean they don't grow in aerobic conditions. In fact, they grow better in aerobic conditions than they do in anaerobic. And then when we start adding sugars, I'm going to circle you back to what I talked about earlier. Human pathogens love sugar. They do. They love any nutrient that you give a human pathogen. Is going to exponentially, rapidly grow. They're going to take it and run. And guess what happens to all of the microbiological structure in that soil or that compost? All of the native organisms that are available in that compost at the end of the maturity cycle, they don't know what to do with nutrients. You add molasses to the microbiota in compost, and you're going to give the benefit to all the enterics in that sample, all the fecal coliforms, which may or may not necessarily be pathogenic. You're going to edge out, you're going to give Pseudomonas and all of those organisms an edge. What I'm trying to say here is that when you start to add agricultural tea additives to your compost or your soil or whatever agricultural tea ingredients, you want to evaluate what you're trying to do in your operation, because you're now providing any residual pathogens a chance to grow even better than all the organisms that you probably want to grow and do the good work that you want to have done. So that's just a word of caution and a melding of ideas in my historical, medical, environmental, and food safety background. So I'll just highlight one paper for you. And there are a lot of papers that really show the same thing, just in different ways. And that's the beauty of research. Scientists, they take what they learned and they put a new spin on it and do some more work. And so this is actually one of my first papers that came out, and I used this data to fuel my dissertation, but blended all the concepts together that I talked to you about. When you think about how many organisms are in a good quality compost, and we're talking millions per gram, and you add maybe 10 or 100 cells of E. coli, that is quite a bit different. The population size is different. So you wouldn't expect the E. coli or Salmonella to survive very long. If you have a single cell of E. coli, 500 grams of compost, you would not expect that to survive very long, and I didn't. Okay. In this paper, I had a really elaborate inoculation scheme, and I take a lot of pride in this because it took a long time to figure out. But I wanted to simulate what would happen if you have a good quality compost on your farm and a goose flies over and poops Salmonella on your compost. So now you have a little concentrated drop of Salmonella. I wanted to simulate that. So what I wanted to do was grow Salmonella and E. coli in compost. And I really couldn't. I tried very hard. I tried to re-inoculate good, mature, S-certified compost with human pathogens and put it in the incubator and let it grow. The most I could get was maybe 3 logs at most in like day 2. And then the population of human pathogens would gradually die off because they didn't have any nutrients. They couldn't compete with the native microbiota structure. And I didn't have the tools that we have now, the metagenomics, to understand all this at the time. So I got frustrated. So what I did was steam pasteurized the compost, and I basically eliminated all the native microbiota from that compost. So I did my best to eliminate all the microbiota in that compost. Then I inoculated that compost with E. coli and Salmonella, and I would get exponential growth of those pathogens in that compost. So that was my starting inoculum. I had a big batch of well-defined compost that had 6 or 7 logs of E. coli in 2 separate batches of Salmonella. So that was my starting inoculum. So then I took some tweezers and I took some particles out of that. I might have probably took a gram of that material and then stuck it in the middle of 500 grams of native good compost to try to simulate what would happen if the bird droppings landed on that compost. So now we have a little tiny couple specks of contaminated compost within that larger batch. And by the calculations that I had, that constituted less than 0.2 CFU per gram of pathogens per compost. So this is well below the detection limit of any assay at the time. And for all intents and purposes, I had a starting batch of clean compost because you couldn't find the pathogens in there. The pathogen content in that starting compost was so low, and the native microbiota was so enormously complex. That was my starting batch. And I used that scheme time and time again. And like all good science, you have to replicate this. And I just kept running these tests because I couldn't believe what was happening. What happened was I had two different types of compost tea. I had aerated tea, and there at the time there was a lot of debate about how aeration eliminated fecal coliform and E. coli populations. And that just didn't happen, and wasn't surprised, but I wanted to run those tests. So in this paper, you're going to see two batches of tea, aerated tea, non-aerated tea. Non-aerated tea was run for 8.5 days. And I had two different additives. At the time. We had a molasses-based additive, which had bat guano in it, as well as a fungal formula, which had stuff like sea kelp, rock dust, fish hydrolysates, and things of that nature. And then we had a no additive tea, which is basically just the compost. And I wanted to make sure there were no issues with water quality. So we used potable water, let it off gas the chlorine for a few days. And there really is only one conclusion that you really need to be aware of. And you can look at the paper and see all the stats, but nutrient supplements clearly were the culprit. Any agricultural tea, whether it aerated or not, had the potential to support rapid growth and sustained human pathogen populations. And so that struck me as really something that we needed to get out. We needed to broadcast that information. And by the way, all of the agricultural tea equipment that I used where nutrients were added, they had an enormous layer of biofilm as well. And you just didn't have that in the non-aerated tea. Of course, every batch was sterilized, all the equipment between reps, so it wasn't an issue. But on the farm, let's face it, growers, cleaning and sanitation is a big issue with a lot of equipment, including agriculture water systems. So this was a big problem for me as a medical microbiologist studying agricultural issues. And I hope this information gets out. This information is buried in subpart F, and I'm going to rip you through a few of the slides that I pulled from our workshops.
Dave.
Yes.
We might end it there on that good keynote about cleaning and sanitizing, unless there's like one or two high-level things you really want to knock out. Otherwise, we might throw the mic and the slides back over to Donna.
1 second, I'm going to share my screen. I just want to talk through really quickly, and I think this will, let me know if you're seeing the wrong screen, but I think this will help answer the question that is being asked. And I think it'll help walk through some of the current FDA guidance that's currently published. So I do want to take a step back, and this is addressing Dave's point about how are these agricultural teas being used. And I think when we work with growers, this is the first question that we try to figure out is, how is it being used? As Dave mentioned, agricultural teas are soil amendments for the purposes of the Produce Safety Rule. And you can see on the right, hopefully it's not too small, there's a screenshot from guidance and from the draft Produce Safety Rule guidance, I should specify. It says, does your farm use soil amendments, including agricultural teas? So ag teas meet that definition of soil amendment. And then how is it being used? So thinking about how it's being used, we talked a little bit about soil drenches earlier, and a soil drench is intentionally added to the soil as an amendment. So I think that very easily meets the definition of a soil amendment. What about foliar application? I think that's where the gray area is. And pulling a figure from guidance, this is what Dave was talking about when he highlighted the two different treatment standards. So there are two standards that are established in the rule. The first one is in yellow, and this is the standard that Billy was mentioning when there's compost, windrow compost. And the requirement there is that it must be applied in a manner that minimizes the potential for contact with covered produce during and after application. So what Dave was just mentioning was the nuclear bomb-proof approach, which is what is in green. So this is almost like a pasteurization step. This might be heat-treated poultry products, poultry pellets. This might pasteurize fish hydrolysate, which is fish emulsion. So it usually goes through some kind of process to show that it has met these additional standards. And that can be applied in any manner with no restrictions. But I think one of the gray area, one of the questions that Dave is highlighting is, what about foliar application of agricultural tea made with like windrow compost? Because according to guidance, that has to be applied in a manner that minimizes the potential. I think Dave did a good job of talking about prioritizing how that is being used, but I want to add in, we're talking a lot about application. And I just want to remind everybody that it's important to also talk about the basic good agricultural practices. So going back to cleaning and sanitizing that came up earlier, that one of the big reasons agricultural tea are used are for that biomass. And a lot of these microorganisms create biofilms. So when the brew equipment is being used, make sure it's being cleaned and sanitized to try to minimize that development of the biofilms, because biofilms can harbor pathogens. Thinking about how it's transporting, trying to designate specific equipment and tools for specifically handling or applying or transporting the soil amendment. And then I'll also put a plug in for worker training if there are specific workers who are doing this, as well as record keeping. So I just wanted to mention that. I thought that was a good reminder of these other good agricultural practices.
Yeah, those are really great.
Thank you all so much. We hope to see you next month.